look at this list of some proteins. You will see that the spectrum of your proteins is very wide. We have the proteinase inhibitor 3, which only has 30 amino acid residues and has a molecular mass of around 3,000 Daltons. In fact, we won't call it a protein, but according to the definition I mentioned in a previous video, this is a polypeptide because its molecular weight is less than 10,000 Daltons. Now, on the other end of the spectrum, we have again titin, which is the largest protein known to date, which this protein has 34,000 plus amino acids. It's uh, 1,000 times the number of amino acid residues of the proteinase inhibitor 3. But although this spectrum is very wide, we see that in this very short list of proteins, majority of them have less than 1,000 amino acids. Now, of course, this is not a comprehensive, exhaustive list of proteins, but we see a pattern. In fact, empirical studies show that you need at least 40 amino acid residues for a polypeptide chain to fold into a discrete, stable shape and executing a certain function. Meanwhile, polypeptides with over 1,000 residues may approach the limit of the protein synthetic machinery of the cell. This means that if the cell tries to produce a lot of proteins with more than 1,000 amino acid residues, it might break down, so to speak. So it doesn't only pose a limit on the synthetic machinery, but it can also be energy demanding. Meanwhile, if we look at the amino acid composition of your proteins, taking these two as representatives, we have cytochrome C and chymotrypsinogen, both from bovine sources. We see that you don't have an equal distribution of your amino acids. In fact, we will see that some amino acids will have very little percentage in the composition of the whole protein. So the 20 common amino acids do not appear with equal frequencies in our proteins. Again, observing your data, most abundant of these amino acids include your leucine, alanine, glycine, valine, glutamate, and serine. Most of these are your nonpolar amino acids, right? The leucine, alanine, and glycine, and also valine. Meanwhile, we have the glutamate, which is negatively charged. The side chain is negatively charged at the physiological pH. It's because for the functions of a lot of proteins, glutamate plays a role, and it's due to its negative charge. And we also have serine. Serine, with its hydroxyl functional group, also plays important roles in the functions of many proteins. Not only does it act as a hydrogen bond donor or acceptor, but it can also act as an acid if it has the proton, or a base if its proton is dissociated. Among these 20 common amino acids, the rarest, or the one with the lowest frequencies of occurrence in your proteins, include tryptophan, cysteine, methionine, and histidine. So if you notice, you have cysteine and methionine there. These are amino acids whose side chains contain sulfur. So probably This might be due to the limit of the amount of sulfur inside our body, which also limits their synthesis inside the body. Now, take note, the character of a protein depends more on its amino acid sequence than on its amino acid composition. So all these comments about the limit and in the size and composition of your proteins are just observations from their amino acid sequences. We are actually more concerned with the character of the protein. So character means their chemical properties and their functions in our biochemical reactions. So this is much like two words having the same letter composition but with different meanings.